Welcome back to the deep dive. You know, when you think about the human body, it's just staggering. Over 200 different kinds of cells, all building these incredibly intricate tissues and organs.
It really is mind-blowing complexity.
But here's something maybe even more amazing that we often just take for granted. Our bodies aren't static at all. They're constantly changing in this state of, dynamic equilibrium.
Yeah, a constant flow. Like a river of cells where new ones are always being born, they specialize, do their job, and then eventually they die off.
Exactly, this constant renewal.
Yeah.
It's how tissues like our skin or our blood or even the lining of our gut manage to stay the same, structurally speaking, even though the individual cells are always turning over. It's quite a balancing act.
It is happening constantly, second by second, inside all of us.
So today we're diving deep into how this incredible renewal works, and we're focusing on the real stars of the show, stem cells.
Yeah.
We'll look at what makes them special, how they keep that balance in our tissues.
And importantly, the huge potential they hold for medicine down the road.
Right. And for this deep dive, we're drawing heavily on Chapter 22 of the classic textbook, Molecular Biology of the Cell, the 7th edition. It's titled Stem Cells in Tissue Homeostasis and Regeneration, and it's just packed with insights into this microscopic world.
Yeah, it's a great source.
Yeah.
So our goal today is really to unpack the core ideas of stem cell biology, get a handle on how these crucial cells are controlled, and maybe get a glimpse of what the future of medicine might look like thanks to stem cell tech.
You should come away with a much better sense of how your own body is constantly rebuilding itself and also what scientists are doing to potentially improve on nature's own designs.
Let's get started.
Okay, so let's start with that idea you mentioned, the river of cells. The book uses this great analogy. A river looks the same day after day, right? But the actual water is always flowing downstream.
But that's tissue homeostasis in a nutshell. Our self-renewing tissues keep their shape and function, but the cells themselves are always being replaced. Old cells are lost downstream.
So the big question is, where do the new ones come from? What's the upstream source keeping everything stable?
That source is the stem cells. They are these specialized, undifferentiated cells. Their whole job is to provide a fresh supply of new cells whenever and wherever they're needed for replacement or maybe repair after injury.
Without them, tissues that turn over quickly, like our gut lining, would just fail, right?
Absolutely. They'd fail rapidly. It's not just some abstract biology concept. It's literally the operating system keeping us going, healing, adapting every single day.
Okay, so what really defines a stem cell? The book highlights two absolutely core properties. First one, self-renewal. What exactly does that mean?
It means a stem cell has this unique ability to divide and make more copies of itself. It essentially replenishes the stem cell pool, usually for the entire life of the organism.
Sort of keeping the source topped up.
Precisely.
Yeah.
Making sure there's always a reserve of stem cells ready to go.
Okay, and the second key property?
That's differentiation. Stem cells can also produce daughter cells that will go on to become specialized mature cells, like a skin cell, a nerve cell, a blood cell.
So when a stem cell divides, its daughters have a choice.
Basically, yes. Each daughter cell essentially decides, do I stay a stem cell and maintain the pool, or do I commit to differentiating and becoming something specialized?
And it's not usually like, poof, you're a skin cell straight from the stem cell, is it? There's often an intermediate step.
That's right, there often is. Stem cells usually generate these intermediate cells called progenitor cells, sometimes called transit-amplifying cells.
Transit-amplifying, okay.
Yeah, these cells are already sort of committed to becoming a specific type of cell, but they divide a limited number of times first. This amplifies the number of specialized cells you get from just one stem cell division. It makes the whole process much more efficient.
Ah, okay, so they ramp up the numbers before the final step.
Exactly. And once a cell reaches the end of that pathway, stops dividing, and is fully specialized, we call it terminally differentiated.
Got it. And you mentioned earlier that stem cells aren't all the same in terms of what they can become. Some are more versatile.
Correct. They can be multipotent, meaning one stem cell can generate multiple different types of specialized cells within a tissue. Or they can be unipotent, meaning they only produce one specific type of differentiated cell. This whole hierarchy, stem cell to progenitor to differentiated cell, is fundamental to how tissues work.
Okay, let's make this more concrete. The book gives some great real-world examples. First up, the lining of our small intestine. It's just one cell thick, but incredibly active. You've got the villi, those finger-like things for absorption.
And then the crypts, which are like these pits dipping down into the tissue below.
Right, so how does the river of cells flow here?
Well, the action, the cell division, happens down in those crypts. That's where the stem cells and progenitor cells live. Then, the differentiated cells migrate upwards out of the crypts and onto the surfaces of the villi. It's a continuous stream.
And what kinds of specialized cells are we talking about?
There are several key players. You have the absorptive cells, obviously, for soaking up nutrients, goblet cells, which secrete the mucus that protects the lining. And then there are Paneth cells.
Ah, yes, you mentioned those. They're down in the crypt base.
Right at the bottom. And they're really interesting because they actually secrete signals that help maintain the stem cells nearby. They're part of the stem cell support system. It's niche. There are also neuroendocrine cells secreting hormones.
So it's literally like a conveyor belt. Cells are born in the crypts, move up, do their job on the villi, and then what happens?
They reach the tip of the villus and are basically shed into the gut lumen. This whole journey for absorptive and goblet cells takes only about 3 to 4 days in a mouse. They're replaced incredibly quickly.
Wow, but the Paneth cells are different.
They are. They live much longer, weeks, right there at the crypt base. The actual stem cells, the ones driving all this, are located just above the Paneth cells. They express a specific marker protein called LGR5.
LGR5.
Yeah. These LGR5 positive cells divide super frequently, like every 24 hours in mice, and they are multipotent. They can generate all the other cell types in the gut lining. It's an amazing piece of biological engineering.
Truly incredible. Okay, from the gut, let's switch gears to something maybe more familiar, our skin. The epidermis. It's quite different structurally.
Very different. Unlike the gut's single layer, the epidermis is multi-layered or stratified. Think of it like layers of bricks.
So where are the stem cells here?
They're in the basal layer, the innermost layer, right next to the underlying connective tissue. These basal stem cells continuously produce progenitor cells, those transit-amplifying cells again.
And these progenitors move outwards.
Exactly. As they move away from the basal layer, they stop dividing and start terminally differentiating. They become these flattened, dead cells packed with keratin called squames. These eventually reach the surface and are shed.
So all that dry skin we slake off, that's the end product of this constant renewal process.
That's precisely it, the visible part of this invisible ongoing construction project.
So even though the gut and skin look totally different, the basic principles are the same. Stem cells hang out in a specific spot, get signals from their environment.
Right, like the basal lamina in the skin or the Paneth cells in the gut.
And they produce these progenitor cells that boost the numbers before the cells finally differentiate and are eventually lost.
You got it. That's the core logic.
Okay, finding these stem cells sounds tricky, though. You said they're often rare and don't necessarily look different. So how do scientists actually track them down and prove they are stem cells?
Yeah, it's a major challenge. One really elegant technique is cell lineage tracing. This uses genetic engineering in animals, usually mice.
How does it work?
Basically, you engineer the animal so you can permanently tag a specific cell and all of its descendants with a marker, like a fluorescent protein, say GFP. You can control when this tagging happens, often using a drug like tamoxifen.
Okay, so you tag a cell, then what?
Then you wait and see what happens. If the cell you happen to tag was a stem cell, you'll see this expanding clone, this patch of marked cells that persists over time. And crucially, this patch will contain both new stem cells, keeping the lineage going, and all the different types of differentiated cells that stem cell can produce.
Ah, so it traces the entire family tree originating from that one cell.
Exactly. It shows you its long-term contribution to the tissue.
And if you tagged a progenitor cell instead?
Then the patch of labeled cells would be transient. It might expand for a bit as the progenitor divides, but eventually all the cells would differentiate and be lost according to the tissue's normal turnover. The label would disappear because progenitor cells don't self-renew indefinitely.
That's really clever. And you can even target the tagging.
Yes. If you know a gene that's only active in the stem cells, like that LGR5 gene in the gut stem cells, you can set up the system so the tag only gets activated in LGR5 expressing cells. When you do that and see a long-lasting clone with all the gut cell types, it's definitive proof that those LGR5 cells are indeed the multipotent stem cells.
Brilliant. It's like putting a GPS tracker on a specific cell type. But what about stem cells that aren't always dividing? You hear about quiescent or dormant stem cells.
Right. Not all stem cells are constantly cycling like the gut ones. Some adult stem cells normally hang out in a quiet state called quiescence. They only wake up and start dividing when needed, usually in response to injury or maybe specific growth signals.
Like in our muscles.
Exactly. Skeletal muscle has these satellite cells. There are normally small, non-dividing cells tucked away next to the muscle fibers. But if you injure the muscle, these satellite cells get activated. They start dividing rapidly, and their descendants fuse together to repair the damaged muscle fibers.
But this repair system isn't perfect, especially as we age or in certain diseases.
Unfortunately, no. In conditions like muscular dystrophy, where there's chronic damage, or just with advanced aging, the satellite cells can't keep up. The repair capacity diminishes, and muscle tissue can get progressively replaced by scar tissue, leading to weakness.
Okay, another classic way to identify stem cells, especially historically, was through transplantation experiments. The blood system is the key example here, right? The hematopoietic system.
Yeah, absolutely. It's arguably the most complex and best understood stem cell system in mammals. Hematopoietic stem cells, or HSCs, live in the bone marrow and are responsible for generating all our blood and immune cells, red cells, white cells, platelets, everything.
And the classic experiment.
It involved irradiating mice. High doses of radiation destroy the hematopoietic system, so the mouse can't make new blood cells and will die.
Grim, but necessary for the experiment.
Right. But if you then take bone marrow cells from a healthy, genetically matched donor mouse and inject them into the irradiated mouse,
It gets rescued.
it gets rescued. The injected bone marrow cells contain HSCs, which migrate back to the recipient's bone marrow, settle in, and completely rebuild the entire blood and immune system. This was fundamental proof that these powerful stem cells exist in bone marrow.
And these HSCs are super rare, aren't they?
Incredibly rare. Maybe only 1 in every 50,000 to 100,000 cells in the bone marrow is a true HSC. But get this. Experiments have shown that transplanting even a single HSC can be enough to completely reconstitute the entire blood system of a mouse for its lifetime.
Just one cell. That's unbelievable power.
It really is. They are definitively multipotent, giving rise to all the dozens of different blood cell types, and they self-renew to maintain the HSC pool for life.
So how do these rare HSCs manage to produce the billions of blood cells we need every single day? They don't divide that often, do they?
No, they divide relatively infrequently. The trick, again, is the hierarchy involving progenitor cells. HSCs first produce multipotent progenitor cells, which then divide more rapidly and produce daughter cells that are progressively more restricted in their potential.
Like branching paths.
Exactly. You might have a common myeloid progenitor giving rise to red cells, platelets, and certain white cells, and a common lymphoid progenitor giving rise to lymphocytes. These then divide further, producing progenitors committed to just one lineage, like an erythroblast progenitor for red blood cells. It's a whole cascade of amplification and specialization.
And why is this hierarchy so important? Why not just have the HSCs divide like crazy?
2 main reasons. First, keeping the number of divisions for the actual stem cells low minimizes the wear and tear, the risk of replicative senescence. That's when cells essentially wear out their chromosome ends, telomeres, after too many divisions and stop dividing altogether.
Cellular exhaustion again.
Right. And second, fewer divisions mean fewer chances for potentially dangerous mutations to accumulate in the stem cells themselves. A mutation in an HSC could be catastrophic, potentially leading to leukemia or other cancers, because it would be passed down to a huge number of descendants. The hierarchy limits this risk.
That makes sense. It's a protective strategy. Now, are there tissues that kind of break these stem cell rules? Tissues that renew differently?
Yes, there are exceptions. Pancreatic beta cells, the ones making insulin, don't seem to rely on a dedicated stem cell pool. Instead, existing differentiated beta cells can just divide to make more beta cells. This is obviously super relevant for understanding diabetes.
And the liver. It's famous for regeneration.
The liver is amazing. Hepatocytes, the main liver cells, normally divide very, very slowly. But if you surgically remove a large part of the liver, say 2/3 of a rat's liver, the remaining hepatocytes undergo this incredible burst of proliferation. They can fully regenerate the lost mass in just a couple of weeks.
Wow, so it's the differentiated cells doing the heavy lifting there.
Mostly, yes. Although there's some evidence for liver progenitor cells too, especially in chronic injury. And sometimes cells can even be flexible. Differentiated cells might de-differentiate back to a progenitor state to help repair, like Schwann cells in nerve injury. Or progenitors might revert back to stem cells if the main stem cell pool is damaged, like in the testes. Nature finds ways.
But some tissues just don't have this capacity at all in mammals, right? Once the cells are gone, they're gone.
Sadly, yes. Key examples are the sensory cells in our inner ear, the hair cells needed for hearing, and the photoreceptor cells in our retina needed for vision. We don't have stem cells to replace these if they're damaged by loud noise, disease, or aging. That's why hearing loss and certain types of blindness are often permanent. It really highlights the need for stem cell therapies in these areas.
Yeah, absolutely. Okay, so we've established stem cells exist and how we find them. Let's talk about control. Stem cells don't just operate in a vacuum, right? Their environment is crucial.
Absolutely critical. Stem cells reside in and are controlled by a specialized local microenvironment called the stem cell niche.
The niche, like their specific neighborhood.
Exactly. This niche provides essential signals, often secreted proteins like WNT or hedgehog, or direct contact-dependent signals like Notch that maintain the stem cells in their undifferentiated self-renewing state.
So the niche tells the stem cell, stay a stem cell.
Pretty much. These signals are often present only within the very confined space of the niche. If a daughter cell produced by stem cell division happens to get pushed outside the niche, it loses those maintenance signals.
And it's forced to differentiate.
Right. It's a simple but effective way to ensure that stem cell numbers are controlled and that differentiation happens when and where it's needed. The physical boundaries of the niche itself limit the stem cell population.
Let's go back to the intestinal crypt. That's a great example of a niche, isn't it?
It's a classic example. We mentioned the Paneth cells at the base. They act as key niche supporting cells. They secrete WNT proteins, which are vital for stimulating stem cell self-renewal. They also have proteins on their surface, like delta, that activate Notch signaling in the stem cells they touch. And Notch signaling tells the stem cell, don't differentiate yet.
So it's this combination of signals from the Paneth cells keeping the LGR5 stem cells right above them in their stem cell state.
Precisely. It's a very well-defined system.
And understanding these niche signals has led to some really cool lab work, right? Like growing mini guts.
Yeah, it's incredible. Scientists figured out the key signals provided by the niche. They found that if you take just a single LGR5 expressing stem cell from a mouse gut and put it in a culture dish with the right mix of growth factors mimicking those niche signals,
What happens?
it starts dividing and self-organizes into a complex, three-dimensional structure that looks remarkably like a miniature gut lining, complete with crypts and villus-like domains. They call these organoids.
Wow, a mini gut in a dish from one cell.
It shows the power of these niche signals and the intrinsic ability of stem cells to build tissues if given the right instructions. These organoids are now invaluable tools for research.
Okay, so the niche controls stem cell identity and numbers, partly by its limited size. What other ways do tissues regulate the balance between making more stem cells versus making differentiating cells?
Another important mechanism, especially in some systems, is asymmetric stem cell division.
Asymmetric, meaning unequal.
Exactly. The stem cell sets itself up before it divides so that the two daughter cells inherit different components, different cell fate determinants. One daughter gets the stuff needed to remain a stem cell, while the other gets signals that push it towards differentiation.
So it guarantees one replacement stem cell and one differentiating cell with each division.
Right. It's a very precise way to maintain the stem cell pool size. A great example is in the Drosophila, fruit fly testis. The stem cell orients itself so that when it divides, one daughter stays attached to the niche cells while the other is pushed away, ensuring their different fates.
That sounds quite deterministic, quite rigid. Is that how it always works?
Not always. Many tissues, especially vertebrates like us, seem to rely more on a flexible strategy, often called symmetric division with independent or stochastic choice.
Stochastic meaning random.
Kind of, or probabilistic. Here, a stem cell might divide symmetrically, producing two initially identical daughters. Each daughter then independently decides, based on local signals or perhaps internal fluctuations, whether to remain a stem cell or differentiate. It's less rigidly controlled than asymmetric division.
Why would that be useful?
It allows for more flexibility. For instance, after an injury, the tissue might need to rapidly expand the stem cell pool. This stochastic system allows for divisions that produce two stem cells. Or, if conditions require more differentiated cells, it can bias towards divisions producing two differentiating cells. The intestinal epithelium seems to use this flexible strategy heavily.
Okay, that makes sense for adapting to changing needs. Now, what happens to all this as we get older? Does stem cell function decline with age?
Unfortunately, it generally does. The ability of our adult stem cells to maintain tissues and respond to injury often decreases with age, and this is thought to be a major contributor to the aging process in many tissues.
How do we know this?
Well, experiments like serial transplantation with hematopoietic stem cells show this clearly. If you take HSCs from a mouse, transplant them into an irradiated recipient, let the blood system recover, then take HSCs from that mouse and transplant them into another irradiated recipient and repeat this.
The HSCs eventually poop out.
Essentially, yes. Their ability to repopulate the blood system diminishes with each transfer. They undergo that replicative senescence we talked about. They have a finite capacity for self-renewal.
So is the problem just the stem cells themselves getting old, or is it their environment, the niche, that's changing, or maybe something systemic in the whole body?
That's the million-dollar question. And a technique called parabiosis helps tease this apart.
Parabiosis, what's that?
It involves surgically joining the circulatory systems of two animals, typically mice. So you might join an old mouse to a young mouse, so they share the same blood supply.
Whoa. Okay, what does that tell you?
It helps distinguish between factors intrinsic to the stem cells versus factors circulating in the blood. And the results are fascinating. Studies have shown that factors in the blood of old mice can actually impair stem cell function, like neurogenesis in the brain, when circulating in a young mouse. It makes the young mouse's system act older.
So old blood is bad for young stem cells.
It seems that way, yes. But the flip side is even more exciting. Factors in the blood of young mice can rejuvenate stem cell function in old mice. They've seen this across various tissues, muscle, brain, liver.
Young blood can make old stem cells act younger.
That's what the evidence suggests. It implies that circulating factors play a significant role in stem cell aging, and that targeting these factors could be a potential avenue for anti-aging therapies. It's a very hot area of research right now.
Absolutely fascinating. Okay, so our bodies do a lot of natural repair, but as we've discussed, there are limits, especially in humans. We can't regrow a whole limb if we lose one.
No, unfortunately not. Our regenerative capacity for complex structures like limbs, or even for tissues like heart muscle after a heart attack or neurons after major injury, is pretty limited compared to some other animals.
Which brings us to nature's regeneration superstars. The book mentions planarian flatworms.
Ah, yes, Schmitty Mediterranea. These things are incredible. They're small, simple worms, but you can cut one into tiny pieces, and almost any piece can regenerate into a complete, perfectly proportioned new worm within days.
From just a fragment.
Even from potentially a single cell. They have this amazing population of adult stem cells called neoblasts distributed throughout their body. These neoblasts are thought to be truly totipotent, capable of making every single cell type in the worm, including germ cells. They are constantly active, replacing cells even during starvation, when the worm shrinks, and driving regrowth when food is available.
That is just astonishing regenerative power, almost alien.
It really is. And then you have amphibians like salamanders, newts, and axolotls.
Regrow limbs,
Yeah, entire limbs, yes. And also tails, jaws, spinal cord, parts of the eye, even brain tissue. If you amputate a salamander's limb, cells near the wound site de-differentiate. They kind of go backwards developmentally and form a structure called a blastema.
A blastema.
It's a clump of rapidly dividing, undifferentiated cells. But crucially, these cells seem to retain some memory of what they originally were and, importantly, their positional identity, whether they came from the upper arm or lower arm, for instance. This allows the blastema to perfectly regenerate the missing part: bone, muscle, nerve, skin, all in the right pattern.
So how do they know where to stop? How do they reform the exact structure?
That's still a major area of research, understanding that positional memory and the signaling involved. But it raises the profound question: why can a salamander regenerate a limb so perfectly, while a mammal forms, well, mostly scar tissue? What's the difference? Unlocking that could be huge.
Absolutely. So given what we do know about our own stem cells, how are we actually using them in medicine today?
Well, we're already leveraging some of our natural stem cell systems. Hematopoietic stem cell transplantation, or bone marrow transplant, is a standard treatment for many leukemias, lymphomas, and other blood disorders.
We talked about the mouse experiments. This is the human version.
Exactly. You destroy the patient's diseased blood-forming system with chemotherapy or radiation, and then you infuse HSCs, often collected from the patient themselves beforehand, autologous transplant, or from a matched donor, allogeneic transplant, to rebuild a healthy blood system. Using the patient's own cells cleverly avoids the problem of immune rejection.
Okay, that's a major success story. What else?
Skin grafting for severe burns is another established application. If someone has extensive burns, you can take small biopsies of their unburned skin, isolate the epidermal stem cells, and culture them in the lab to grow large sheets of new epidermis.
Grow new skin in the lab.
Essentially, yes. These sheets can then be grafted back onto the patient to cover the burned areas. It uses the skin's own regenerative capacity, just amplified in the lab.
Incredible. What about the nervous system? We said it's tricky, but are there any glimmers of hope using neural stem cells?
There are. Although adult neurogenesis, making new neurons, is limited in mammals, we do have neural stem cells in a couple of specific brain regions, like near the hippocampus, involved in memory, and in the lining of the brain ventricles, supplying neurons to the olfactory bulb.
So there is some natural neuronal turnover.
A little bit, yes. And importantly, scientists can isolate these neural stem cells from fetal or even adult brain tissue and grow them in culture. They often form these floating clumps called neurospheres.
And what can you do with neurospheres?
If you provide the right signals, you can coax them to differentiate into various types of neurons and glial support cells. Even more interestingly, if you transplant these cultured neural stem cells or progenitors back into an animal's brain, even into a different region.
They can survive and integrate.
Yes, they show a remarkable ability to respond to the local environment, differentiate appropriately for that region, and integrate, at least to some extent. This offers real hope, though still largely experimental, for treating neurodegenerative diseases like Parkinson's or repairing damage from stroke or spinal cord injury by replacing lost cells.
Okay, this is leading us towards the truly revolutionary stuff. The idea of actually reprogramming cells, changing their fundamental identity.
Exactly. Can we take an easily accessible cell, like a skin cell, and turn it into something completely different that the body needs, like a neuron or a heart muscle cell?
The early hints came from those nuclear transplantation experiments, right? Like with the frogs and Dolly the sheep.
Precisely. Taking the nucleus from a differentiated cell, like a tadpole gut cell or a sheep mammary cell, and putting it into an egg cell whose own nucleus was removed, showed that the differentiated nucleus still contained all the genetic information needed to make a whole new organism. The egg cytoplasm somehow reset or reprogrammed the nucleus back to an embryonic state.
But it wasn't very efficient, was it?
No, extremely inefficient and often led to developmental abnormalities. It proved the principle, but also highlighted that reprogramming involves massive, complex changes in how genes are controlled. Things like chromatin structure and DNA methylation patterns need a total overhaul.
So cloning wasn't the therapeutic answer. But then came embryonic stem, ES cells.
Right, these were first isolated from mouse embryos and later human embryos, specifically from the inner cell mass of the blastocyst, that very early stage embryo.
And what's so special about ES cells?
Their defining feature is pluripotency. They can divide indefinitely in culture, maintaining an undifferentiated state, and they retain the potential to differentiate into essentially any cell type in the adult body. They are the ultimate flexible building block.
And they were huge for research.
Absolutely transformative, especially in mice. ES cells allowed scientists to make precise genetic modifications and then generate knockout or knock-in mice, revolutionizing our ability to study gene function in development and disease. Human ES cells offer the promise of an unlimited source of cells for potential therapies.
How do they keep dividing forever without wearing out like normal cells?
They express high levels of an enzyme called telomerase, which maintains the protective caps, telomeres, at the ends of chromosomes. Normal cells usually lose a bit of telomere with each division, eventually triggering senescence, but ES cells keep their telomeres long. Their pluripotent state is actively maintained by a core network of transcription factors, like octave4.
Okay, ES cells were amazing, but using cells derived from embryos raised ethical concerns and also faced the issue of immune rejection if transplanted. Then came the real game changer.
Induced pluripotent stem, iPS cells. This was the bombshell discovery in 2006 by Shinya Yamanaka's lab.
What did they do?
They showed that you could take ordinary differentiated adult cells. They started with mouse fibroblasts, skin cells, and turned them back to cells that were essentially identical to ES cells just by artificially introducing a small set of specific transcription factors.
Turning the clock back on a differentiated cell, which factors were key?
The core cocktail became known as the OSKM factors: OCT4, SOX2, KLF4, and MyNIC. Just forcing the expression of these four master regulators was enough to induce reprogramming.
So you could take your own skin cell, turn it into an iPS cell, and now you have pluripotent stem cells that are genetically identical to you.
Exactly. That was the revolution. iPS cells share all the key properties of ES cells: indefinite self-renewal, pluripotency. They can form all cell types and contribute to chimeric animals, but they can be made from any individual. This bypasses the ethical concerns around embryos and, crucially, solves the immune rejection problem for potential transplantation therapies.
It sounds almost too simple, just add four factors, but the book emphasizes it's actually a really complex and inefficient process.
Oh, absolutely. It's not like flipping a switch. Reprogramming is typically very slow, taking weeks, and only a tiny fraction of the starting cells actually succeed in becoming iPS cells. It requires what the book calls a massive upheaval of the entire gene control system.
Not just turning on those four genes.
No, those four factors initiate a cascade that leads to widespread changes in chromatin structure, DNA methylation, histone modifications. The whole epigenetic landscape has to be reset from a differentiated state back to a pluripotent one. It's a messy stochastic process.
And scientists figured out how to find those few successful cells.
Yeah, clever selection strategies were key. For example, linking the expression of a drug resistance gene to a pluripotency marker gene, so only the fully reprogrammed cells could survive when the drug was added.
And they're even finding ways to make it more efficient now.
Yes, by understanding the molecular barriers to reprogramming. Manipulating other proteins involved in chromatin remodeling or histone modification can significantly boost the efficiency, sometimes by orders of magnitude. It shows how understanding the basic mechanisms of gene control is crucial for advancing these technologies.
Okay, so now we have these powerful pluripotent cells, ES or iPS. How do we actually use them for therapies? You can't just inject them straight into someone, right? You mentioned teratomas.
Correct. If you inject undifferentiated pluripotent cells directly into an adult tissue, they tend to form teratomas. These are usually benign tumors, but they contain a chaotic mix of different tissues, bits of skin, hair, muscle, even teeth, because the cells differentiate randomly without the proper developmental cues.
So you need to guide them first.
Exactly. The key is to direct their differentiation in culture before transplantation. By exposing the ES or iPS cells to a specific sequence of signaling molecules and growth factors, mimicking the signals they would normally see during embryonic development, you can coax them down a specific path.
Towards becoming, say, heart muscle cells.
Or dopamine-producing neurons for Parkinson's disease, or insulin-producing beta cells for diabetes. Researchers had developed detailed protocols, like recipes, to generate large populations of specific desired cell types from pluripotent precursors.
And this also leads back to those amazing organoids.
Yes. The ability of ES and iPS cells to respond to signals and self-organize is astonishing. In culture, under the right conditions, they don't just make specific cell types. They can assemble themselves into complex, three-dimensional structures resembling miniature organoids. We talked about gut organoids, but people have made many brains, many kidneys, optic cups that resemble early eyes.
Mini organs in a dish. What are they useful for?
They are incredible tools for studying normal human development, modeling diseases in a human context, and testing drug responses, all without needing animal models or invasive procedures on patients.
Okay, so we can go from differentiated cell back to pluripotent iPS cell, then forward again to a specific differentiated cell type. Is there an even shorter route? Can we go directly from one differentiated cell type to another?
Yes, that's the idea behind direct transdifferentiation or direct reprogramming. The goal is to convert, say, a fibroblast directly into a neuron or the liver cell without going through the pluripotent iPS stage.
Skipping the middleman? How does that work?
It also relies on introducing specific master transcription regulators, but a different set ones that are characteristic of the target cell type. For example, researchers found a combination of factors that could convert fibroblasts directly into functional neurons.
And the heart repair example you mentioned earlier?
That's a really exciting one. Scientists identified 3 transcription factors, GATA4, MEF2C, TB5, that when introduced into heart fibroblasts, the cells that normally form scar tissue after a heart attack, could directly convert them into beating heart muscle cells, right there in the damaged heart tissue in animal models.
Convert scar tissue into functional muscle. That would be revolutionary for heart attack patients.
It absolutely would.
Yeah.
It's still experimental, but it shows the potential of reprogramming cells in vivo to repair damage.
Amazing potential. So putting it all together, especially with iPS cells, what are the biggest impacts right now? Is it mainly transplantation therapies?
Transplantation is the long-term goal, and solving the immune rejection issue with patient-specific iPS cells is a huge step. But arguably, the most immediate and impactful application of iPS technology currently is in disease modeling and drug discovery.
How does that work in practice?
You take cells, say skin cells, from a patient with a specific genetic disease. You make iPS cells from them. Now you have an unlimited supply of pluripotent cells carrying that patient's disease-causing mutation. Then you differentiate those iPS cells into the cell type that's primarily affected by the disease, maybe neurons for a neurological disorder, or heart cells for a cardiac condition.
So you have the patient's disease in a dish.
Essentially, yes. You can study exactly how the mutation affects the cell's function,
Yeah.
what goes wrong at a molecular level, and importantly, you can use these disease cells as a platform to screen thousands of potential drug compounds to see if any can correct the defect or alleviate the symptoms.
Like the Timothy syndrome example.
Exactly. Researchers made iPS-derived heart cells from Timothy syndrome patients. These cells showed the characteristic irregular electrical activity and contractions seen in the patients. They could then test drugs directly on these cells to find ones that normalize the heart rhythm. It's a powerful way to understand disease and find treatments tailored to human genetics.
And the microcephaly example using brain organoids.
Similar principle. Brain organoids grown from iPS cells of a patient with a specific form of microcephaly revealed that the neural progenitor cells were stopping division too early, leading to a smaller brain. This provided crucial insight into the disease mechanism that would have been impossible to get otherwise.
Wow. It really feels like we've covered a vast landscape today, from the fundamental daily renewal in our tissues, this constant cellular dance.
All the way to these incredible futuristic possibilities of reprogramming cells to fix damage, model diseases, and potentially cure conditions we couldn't touch before.
It's just amazing how far our understanding of stem cells has come. It really offers completely new ways to think about health, disease, and aging.
It truly does. This field unveils not just the body's inherent resilience, but also the incredible power we're starting to harness through science to potentially enhance that resilience or restore function when it's lost.
So, thinking ahead, as we get closer to a future where maybe we can reliably mend broken hearts with new muscle cells growing from skin, or maybe even regrow damaged nerves, what does that mean for us? How does it change our definition of repair, or even what it means to be human, if we can essentially reset and rebuild parts of ourselves at this fundamental cellular level?
That's a deep question, isn't it? It pushes the boundaries of medicine into philosophy almost. What are the limits? What should the limits be? It's a conversation that science and society will need to have as these technologies continue to mature. It makes you really ponder what other secrets our cells hold.
Definitely food for thought. Well, thank you for joining us on this deep dive into the absolutely fascinating world of stem cells and regeneration. We really appreciate you being part of our deep dive family.
Always a pleasure to explore this stuff.
Until next time, keep your curiosity alive.